My name is Stephanie Hicks. I am a faculty member at Johns Hopkins. Actually, a little bit about me. I've loved that everybody started with the About Me slides. It's very helpful. So I am a faculty member at Johns Hopkins in the Biostats department, similar to Roger Pang. I have a couple interests. One is data science education. So if you have any interest in talking about that, I would love to talk to you about that. That's not what I'm going to talk about today, but I have a lot of opinions about that. What I am going to talk about today is specifically a little bit about what I do for my research. So my research is in the analysis of genomic data. Who knows what genomic data is? Okay, that's a great number of you. Awesome. So if you do work with genomic data, you may or may not have heard of the software project called Bioconductor. I'm going to explain a little bit about what that is. It's kind of like the cousin or sister project to CRAN, specifically for the analysis of genomic data. And a few other fun facts about myself. I recently founded the Our Ladies Baltimore chapter as of May 2018, so we are on our third event next week. But I've loved getting to meet so many Our Ladies here in the local DC area. And then I'm also creating a children's book for featuring women statisticians and data scientists. So as you know, I have, or as Jared mentioned, they have young kids, and I went on a mission to try and find children's books featuring statisticians and data scientists. There weren't really any. There were some specifically for STEM, but I'm on a mission to make that happen for statisticians and data scientists. So I'm going to give you a little bit of a taste of that at the end of the talk. All right, another thing about myself, as Jared mentioned, I have two young kids. Theo is my youngest. Happy birthday, Theo. He was born a year ago yesterday. And Alex is my oldest. So he's 2.5, and if you look at this picture long enough, you'll quickly realize two things. One, this was taken very recently. It was the day we voted earlier this week, so he's got his future voter sticker on. And then two, you'll see that he's got minions on his shirt, or as he likes to call them, aliens. And so when I was preparing my talk at the last minute, I'm sure as all of us did, I decided to go with the minion theme. So, CRAN. So as most of us are here for an RStats conference, we all know what CRAN is. It's a great software project that's got over 10,000 R packages to this date, and it does basic things such as data wrangling, or creating plots, but then it also can do more advanced things such as web scraping or analysis of financial time series data or analysis of clinical trial data and so forth. We all love CRAN. So if we look at the subset of people that are in this room, for example, half of us are on our phone either tweeting or looking up cool links or learning about new packages. You might also be on Twitter, and so you might be tweeting about these hashtags, RStatsDC and RStats and Our Ladies. And you can go on your phone or computer to the rproject.org website, and if you go there, there's something called, there's a link called Other Projects. Does this work? Yeah. There's a link called Other Projects, and if you go there, you'll see a few things. There's this thing called Community Services area, which is wonderful, and then right below it is something called Special Areas of Application. So, under the first item under the special areas of application is something called Bioconductor, bioinformatics with R. So it has a list of broad goals. The broad goals are to provide access to a wide range of powerful and statistical graphical methods for the analysis of genomic data, facilitating the integration of biological data, allowing the rapid development that's scalable and interoperable software, promoting high-quality reproducible research, and creating training in computational and statistical methods for the analysis of genomic data. So that gives you a broad overview of what is Bioconductor. So for those of you that have not heard of Bioconductor, formal introductions. CRAN, meet your cool cousin, Bioconductor. Okay, more seriously. Bioconductor is an open source and open development software project, meaning open source, anybody can go look at the code, anybody can go use the code. Open development, meaning anybody can contribute code. Began in 2001 under Robert Gentleman from Genentech, now at 23andMe, I believe. And now it's, today, I think it's got over 12 full-time employees that are funded by the Bioconductor project that work on developing the core infrastructure and the maintenance of the packages. And Bioconductor has some big priorities. I would argue that those are reproducible research and high quality documentation. So for example, every software package in Bioconductor comes with a vignette, and that is really helpful when you don't necessarily want to scroll through just the reference manual. The vignette is meant to walk you through an example analysis or to demonstrate actually how to use the package, which is not the same thing of just looking up a function inside the reference manual. It has a pretty diverse community support group. So we have an online forum that you can go ask questions and you can get experts to respond to your questions. We also have a Slack team that you can join and you can ask those questions in the Slack environment. Workflows. So Bioconductor is really nice because they've developed these users have developed and contributed workflows specific for different types of genomic analysis. So if you've ever heard of the analysis of gene expression data or DNA methylation data or mutations in your genome, there are workflows. If you are new to the field of genomics and you want to figure out how do I analyze my fancy genomic data, you can just go download a workflow and follow the steps that they have demonstrated for you to analyze your own data. And then teaching resources. So Bioconductor has spent a lot of time and effort developing teaching resources that you can use in the classroom to teach your students. So when you think about Bioconductor, the image that Bioconductor aims for is that when you are analyzing a lot of genomic data, there are a lot of great and unique packages out there and tools that you can use to analyze the data, but they're very unique, similar to a musician. A musician is very unique, and Bioconductor serves as the conductor of the set of musicians in an orchestra to keep things on pace and make sure everything works together in a flexible way. Okay, so in the next part of my talk, I'm going to give you a quick demonstration of a little bit about Bioconductor, like what's in Bioconductor, and then a package that I think you'll find really useful. So first is a package called BioC Package Tools, and this contains functions that assess metadata around Bioconductor packages, and it's in a tidy data format. So if you load in the library packages, or library BioC Package Tools, there's one function, BioC download stats, and in one line of code you can have a tibble that's loaded into R with a row containing information with the name of the package, the number of distinct downloads IPs for a given month and year, and there's this last column called repo. So this is the type of package it is. So you could ask, well, how many types of packages are there? So for example, because this is a tibble, then the world of TidyVerse is open up to you. So you can apply anything you want in the TidyVerse here. So for example, you can use the filter function to select the rows from 2018, select just the package and repo column, ask for the number of distinct rows, group by repo, whatever that is, and then summarize by the total number of packages. And you'll quickly see that Bioconductor has three main types of packages. One is software, similar to the software that's available in CRAN. And then two, we have these things called annotation packages and experiment data packages. And if you've ever done any type of analysis in genomic data, you'll realize there's a lot of bookkeeping that has to be done. So for example, if you want to look at genes that are expressed in the human genome, you have to ask what reference human genome it is. For example, every 6 months to a year, the reference genome changes. So like the position where you would expect a gene to be in one reference genome may be slightly different than the position you expected in a different reference genome. So there's a lot of bookkeeping, and the annotation packages basically streamlines that process and makes your life much easier. The experimental data packages, they contain essentially processed data. That is great for teaching because the data is just kind of ready there for you to go. Yeah. You could also ask, so CRAN, as I mentioned, has over 10,000 packages currently available. Bioconductor has around 1,700, but it's been steadily increasing. So inside of the BioC Package Tools function, it only loads in data from, I think, 2009. I'm not sure why there's no data before that. I know there's data somewhere, but it's not in that function. But starting in 2009, it looked like there were around 400 or 500 packages, and we've been steadily increasing through 2018. And this is just software packages. So we have a really big, diverse community of packages. Okay, So what is the standard object? When I talk about genomic data, how does R think about that? So there's this thing called genomic ranges, and I would argue that that's one of the most standard objects that Bioconductor uses. Genomics ranges is essentially a data frame, a constrained data frame. So you can, up here, there's a chromosome. So this is not a piece of data. This is like, and you can imagine a chromosome in our body, chromosome 1. And then inside of this region right here, this is a genomic location in that chromosome. And so when you translate that to R, or when you translate that to something on the computer, we want to record, for example, in that one specific genomic region, we want to record a few things. One, we want to know what's the chromosome number. So in this case, it's chromosome 1. 2, what's the start of that genomic location? So how many bases in, starting from 1, 2, 3, 4, 5, all the way down, does my genomic region start, and does my genomic region end? In the world of genomics, there's positive strands and negative strands. I'm not going to go into that, but it's, again, bookkeeping, essentially. And then there's this thing, there's a dotted line here. And everything to the dotted line is considered metadata. That can be whatever you want. So on the left side of the table, that is a constrained data frame. A G-ranges object expects those 4 columns every single time you create a G-ranges object. On the right side, you can put whatever you want there. So if you think about this in the world of the TidyVerse, this is actually already a tidy data frame because each row is one observation, namely a specific genomic region, and then every column is a specific variable. So I'm going to explain why that's useful in a minute. So how do you create a genomic ranges object? It's really easy. There's a package called Genomic Ranges, and there's a function called GR ranges. So you can create a genomic ranges object by providing those 4 column names that it was looking for. So seq names contains information about the chromosome location. Strand contains information about the positive or negative direction of the chromosome. And then there's this thing called ranges. Ranges are essentially a simplified version of G ranges. It doesn't have information about the chromosome name. It just contains information about the start and the end of the genomic region. So thinking back to this figure that I was showing you before, it was starting at this position for these 2 genomic regions and ending at this base position. And then we had these 2 pieces of metadata, gene ID or the gene name, you can think about it like that, and score. Score is super generic, but you can just think about it as something that you've measured about that genomic region. So in a really quick way, we've now loaded up a standard infrastructure, I'd argue the standard infrastructure that Bioconductor uses to build almost all of its packages when you're talking about genomic data. Okay, so what can you do with that? I mean, that's great, but what can you do with that? So for example, it's common to ask, what's the width of that genomic region? Like how long or wide is that genomic region? So for example, you are literally subtracting the end value from the beginning value, and you can get the width of that for those 2 genomic regions. You can also do things to select or filter for a set of rows. This is base R notation, so GR, which is the genomic ranges object, open bracket. This is the classic base R way to filter a set of rows, looking for all the genomic ranges that have a score greater than 15. And so, similar to the reasons that the TidyVerse and dplyr was invented, that's not superhuman readable, and so there has been recent efforts to try and make the analysis of genomic data more human readable, specifically with what I have discovered recently called the Ply Ranges package. So if you're interested in getting into the analysis of genomic data, I would suggest checking it out because I've incorporated it into my workflow and it's been wonderful. And the whole goal was to just make the genomic data analysis more human readable. So this is not my work. This is the brainchild Stuart Lee from Monash University, Di Cook, and Michael Lawrence. He's one of the he works at Genentech, but he's one of the main core bioconductor developers. And so the idea for this is to define an API, meaning literally extend the dplyr package that maps relational genomic algebra to verbs, similar to the way dplyr does, that act on this tidy genomic data. Another great idea to just straight up borrow dplyr syntax and design principles. And another great idea, compose verbs together with a pipe operator from Magritter. That would make this process of analysis of genomic data much more human readable. So, for example, you can load in the ply ranges object, and instead of doing the base R way to selecting a set of rows, you can take the granges object now, pipe it into the filter function. So for those of you familiar with the dplyr package, you'll recognize the filter function. So you're filtering for the genomic regions that have a score of greater than 15. So we had one genomic region that met that criteria. You can pipe that genomic ranges object that's now filtered into the function called width and ask for the width of that genomic region and so forth. So we're really taking the analysis of genomic data, or the packages that have been developed on the granges object, and making them much more human readable. There is a whole set of verbs that have been developed for the ply ranges package. For example, some of these you'll recognize. The ones in bold were literally, the origin of them came from the dplyr package. So you'll recognize summarize, mutate, select, arrange, group by, and filter. And then the ones that are not in bold are very similar. They're just some unique things to genomic data that make the analysis more fun, as I would like to say. And so they are doing very similar things. Like, for example, there's a whole set of joins and unions. There is a set of filter by overlaps, filter by non-overlaps, and so forth. So, for example, if I were interested in joining two genomic ranges objects, for example, if in the figure A, I have a genomic ranges object X and a genomic ranges object Y depicted by the pink bars. So if I wanted to join those two genomic ranges objects, there are a couple ways I could think about doing that. I could join them by taking the overlap of the intersection, or of the inner part of the genomic ranges object, or the genomic regions, or the intersect, or the left. They're just a lot of different things that are unique to genomic data. And then, because everything is in a tidy framework, again, the world of the TidyVerse is open to you. So, for example, you can make fantastic ggplots and so forth. And that's it. So this is my hat tip to Gabby. So she's the founder of the global organization called Our Ladies, and I mentioned that I'm creating a children's book. So I'm working with a sketch artist who works at Hopkins, and she's been doing a fantastic job. So if anybody's interested in getting involved in this project, I would love your help. I can tell you all about it. And thank you for your time.